It's my honor to introduce our esteemed speaker for the next keynote, Dr. Daphne Koller. She is the CEO and the founder of InSitro, the startup company that aims to rethink drug discovery using machine learning. She is also the co-chair of the board and the co-founder of Coursera. Daphne was the Rajiv Motwani Professor of Computer Science at Stanford University, where she served for 18 years as the faculty member. She also has been the Chief Computing Officer of Calico, an alphabet company in the healthcare space. She is a prolific scientist herself and has over 200 refereed publications. Daphne was recognized as one of the Time's 100 most influential people in 2012 and Newsweek's 10 most important people in 2010. In her exemplary career, she has been awarded multiple awards and fellowships, including the Sloan Foundation Faculty Fellowship in 1996, the ONR Young Investigator Award in 1998, the Presidential Early Career Award for Scientists and Engineers in 1999, the IJCAI Computers and Thought Award in 2001, the MacArthur Foundation Fellowship in 2004, the ACM Prize in Computing in 2008. She was inducted into the National Academy of Engineering in 2011, and elected a Fellow of the American Academy of Arts and Sciences in 2014, and of the International Society of Computational Biology in 2017. Last but not the least, she is an educator par excellence. Her teaching was recognized via Stanford Medal for Excellence in Fostering Undergraduate Research, and as a Bass University Fellow in Undergraduate Education. I'm very thrilled to have her here as the keynote speaker, and I'm looking forward to an inspiring and enlightening talk. So without much further ado, welcome Daphne Koller.
Thank you. It's a pleasure to be here, and it's wonderful to see such an engaged and active community for machine learning in the Bay Area, so thank you all for being here. So today I'm going to tell you about an area that's really been sort of something of a focus of mine for for a couple decades now, which is the intersection between machine learning and the life sciences. And you could say that I've actually been working at the boundary of those two disciplines since around 2000. And I would like to say that I've been working at the intersection of the two disciplines, but there really wasn't that much of an intersection because big data in biology used to be 100 samples. And so you couldn't really do much machine learning with that. That day has changed, and now we're in an era where you could actually do meaningful, exciting machine learning in the biomedical space. So within that space, there's many cool areas that one can focus on, and I've chosen, for reasons that I'll explain in a moment, to focus on how we can make drugs better, faster, and cheaper. So in some respects, you could say we're already there. There's been such a wealth of amazing advances in medicine in the last few decades. Vaccines, biologic therapeutics, antibodies that help with everything from cancer to autoimmune diseases, cancer immunotherapies, the targeted small molecule therapies like for cystic fibrosis that really target very specific lesions on a gene and turn the lifespan of a cystic fibrosis patient from 20 years to almost a normal lifespan. And more recently, the approval of cell therapies, where cells themselves are therapeutic agents. So this is an amazing amount of progress. So this is the glass half full version. The glass half empty is this one. It's a graph that's come to be known as Eroom's law. So Eroom's law, if you notice, is the inversion of Moore's law. Moore's law, which all of us have been living with or riding the tidal wave of, is the exponential increase in productivity of technology. Eroom's law is the exponential decrease in productivity of pharmaceutical R&D. You'll notice that the graph is on a logarithmic scale, and consistently for the last 70 years, the number of drugs approved per US dollars has been decreasing exponentially year on year, despite all of these amazing advances that I showed in the previous slide. So why is that? Why is it that in the 1970s it cost maybe $100 or $200 million to create a new drug, and today it costs over $2.5 billion to create a drug? It is not because the cost of taking one molecule beginning to end has increased by a factor of 25. I mean, yes, there has been an increase in cost, but it's not been that dramatic. The reason is that the number of hypotheses that we need to explore for every drug that succeeds is incredibly large. So many things fail at every single step of pharmaceutical R&D, from the very earliest preclinical phases all the way to approval. So when you think about what that means, this is fundamentally a problem of prediction. So if we were able, at an earlier stage, to say, you know what, we shouldn't bother with these candidates. They are unlikely to pan out. Let's focus on a much smaller set. Then one successful drug would not then have to carry on its back the costs of of so many hypotheses that failed, and we would be able, hopefully, bend that curve on the previous slide and reduce the cost of each of those drugs that we finally bring to market. So at In Citro, we are driven by the philosophy that the prediction is going to bend that curve. And we believe that if you actually train models to make good predictions versus using rules of thumb, historical heuristics, or simply gut instinct, that will be transformative. So we're thinking about the entire R&D value chain and looking for areas where if we only had a good predictive model, it would actually make a very big difference. That machine learning, given the right data, can build a good predictive model. And the right data is really important because one of the main observations after 20 years of being in this space is that the data that you scrape off of people's supplementary materials in reading papers in biology, that data is not good enough. It's often poor quality, and even when it's not fit to purpose. It wasn't designed with machine learning in mind. And so the third and really important bullet is that we're looking for areas where there are technologies that can produce the right data at high quality and at sufficient scale, and we're prepared to then make the investment of actually building the experimental platforms that will generate the data to drive machine learning models. So the company is really half wet, half dry. Half of the company is high throughput biologists who are generating data for the purpose of driving machine learning models, and half the company is machine learning scientists and engineers who are building machine learning models with those data and using the insights to then drive back to the production of additional data using what you might think of as a closed loop active learning, and I'll talk a little bit more about that at the end. Okay, so there's many problems like that in the pharma R&D chain, and I'm not going to talk about all of them today. We haven't tackled all of them, and we aren't talking about the ones that we have. But the first problem that we are tackling is, I think, at the heart of what makes most drugs fail, which is we cannot predict what an intervention do when we administer it to a person, or even more so, to a particular person. So if I put this drug in a human, what will it do is a really fundamental problem, and most drugs fail because of lack of efficacy. So right now, this is typically done using a model system because you don't get to experiment in humans, except for that very limited clinical trial phase, which is very small, typically, and very regimented. And so people do all of their experimentation in model systems, and oftentimes those are mice. And so you take mice, typically actually isogenic mice strains, like all of them are for exactly the same strain, and then you do the experiment in a mouse. You say, oh, the mouse looks like it's better, and so I'm going to then try it in a human. The fundamental problem with that is that mice are not people. And many of the mouse models that we have for disease are actually incredibly badly broken. So I'm just going to give you one of my favorite examples of a mouse model for disease just to show you how bad it can get. It's the mouse model for depression. It's called the forced swim test. You take a mouse, you put it in a bucket, and you force it to swim until it gets tired and drowns. And if it swims longer, it's less depressed. Okay? So when you think about that, it kind of explains why we have so few psychiatric drugs. So I'm not the first to make that observation that mice are not people, and there is a growing move within the community to move to humans as a model for humans. And since we don't get to make experiments in humans, we look to experiments of nature to give us some intuition about what perturbations in humans actually do to human phenotypes, to human traits. And sure enough, it turns out that if you look for cases where nature has perturbed a particular gene and that drives a particular phenotype in the human, drugs that target those genes do considerably better in clinical trials than drugs that do not have that genetic support. So if you look at the odds ratio of approval for drugs that do have genetic support versus drugs that don't have genetic support, and you look all the way through the value chain, which would be the graph on the right, you can see that the approval odds are more than 2. Relative to, so drugs are more than twice as likely to be approved if they have human genetic evidence that backs the relevance of the target to the clinical outcome than drugs that don't have that. So that's great. So the good news is that we have a lot of data that can help support this type of analysis. So this is actually a Moore's law graph as opposed to an Irum's law graph. You can see, again, logarithmic scale. This is the number of human genomes that have been sequenced since the very first human genome in 2001. And you can see that not only is that number growing exponentially fast, it's growing twice as fast as Moore's law. So depending on whether you believe that trend line to continue or to slow down so that it's only as fast as Moore's law, the number of human genomes that will have sequenced by, say, 2025 is between 100 million and 2 billion. Either way, this is a very large number. So that's great. And now, of course, that's useful in and of itself, but it's even more useful if you juxtapose it with clinically relevant human phenotypes. Those, unfortunately, are a little harder to come by. They require a lot of work to collect, at least today. But there is an increasing number of repositories from biobanks where people have actually created aligned data sets of genotypes to phenotypes. One of the best used ones is the UK Biobank. They were early to this game, and they took 500,000 people, very diverse. In addition to measuring their genetics, they also measured a whole slew of phenotypes that are both clinical, as well as environmental, imaging, biomarkers in the blood and in the urine. I mean, the number is there's 4,000 phenotypes or more, depending on how you count in this data set. And there's more such repositories coming up, both national repositories, including here in the United States. There is the emerging All of Us data set, which is going to be 1 million individuals, and multiple other countries and other organizations are doing similar things. So that's great. And now you can ask yourself the question. We have genetics and the variation in the genome. We have the phenotypes. And now you can ask yourself the machine learning question of, can I predict or associate one with the other? And that suggests causality, because fundamentally the genome comes first. And so if you see an association between the genetics and the outcomes, that at least provides considerable support for a causal connection. There's a lot of ifs, ands, and buts on that, which I can speak to later. But fundamentally, there's very strong rationale then for saying something about the causal nature of the target relative to the phenotype. So that's great, and sure enough, if you go back to you go back to 2008, which was the earliest what's called genome-wide association studies, you can see that over the past decade or so, there have been a very large number of associations between genotypes and phenotypes that include diseases, Crohn's disease, Alzheimer's, many others, include various biomarkers like QT intervals or cholesterol levels and so on and so forth, and even things that are not clinical, but things like educational attainment also have a relatively significant genetic basis. So this is great, and you think, well, okay, we're done. We have the causal connection between the genotypes and the phenotypes. Those are our drug targets, right? Well, life is not that simple. Because despite the fact that we have an increasing number of these genetic associations, many of these are really unclear. They do not sit inside genes, they sit outside genes, often in areas that we don't even know what those areas do in terms of their function. Many times, at least as seen across a population-wide level, the effect sizes on the traits can be quite small. And so you don't really know which of the hundreds of Alzheimer's disease loci in the genome are the ones that you would actually go after. And given that each of those drug discovery projects is tens of millions, hundreds of millions of dollars, you can't go after all of them. And so how do you figure out which ones to go after? And sure enough, it turns out that that makes a very big difference. So this is the graph that I showed a few slides ago. And if you now break that down, this 2.0 odds ratio into two categories, the so-called Mendelian traits, where you have a clear, obvious association between the genetic variant and the phenotype versus stuff that comes out of these broad genome-wide association studies, you see that for the Mendelian traits, the odds ratio is greater than 2.5, whereas for the GWAS hits, it's less than 1.5. So there's still a signal there, but it's not as readily interpretable. And so it's not that easy to go from those broad genome-wide association studies, especially for those complex traits like Alzheimer's or schizophrenia, to things that are actionable drug targets. So how do we do this differently? Is there another source of data that we could use, given that fundamentally human data is very limited in terms of our ability to especially experiment on humans is a very limited thing that we could do. So this is where we're going to leverage another revolution, which is the ability to generate biological molecular data at unprecedented fidelity and scale. This, too, is a revolution that's come about in the last few years. And because it is something that gets you closer to the underlying causal biology, it allows you to bridge the gap between the genetics and the phenotype, and also, frankly, considerably increase the amount of data that is available to us in doing machine learning. So let me give an example of what I mean here, and then we'll talk about the data generation process. This is a case study in CNS disease, central nervous system. There is a region in chromosome 16. It's called 16p11.2. For whatever reason, that region is subject to copy number changes in humans. It's deleted in some individuals and duplicated in others relative to wild type. It turns out that when it's deleted, you have an over 75% penetrance for autism. And when it's duplicated, you have an approximately 40% penetrance for schizophrenia. No one knows exactly why. There's 25 genes in that region and sundry other elements, promoters, enhancers, all sorts of other things. Which of those, or combination of those, causes this phenotype, and why does it happen? Not clear. So 2 years ago, a group, Deshpande et al., in a paper in Cell Reports, basically said, well, let's figure it out. So what they did, and this is where the magic of some of the technologies that have emerged in the last few years can really come about, they took skin cells from a number of patients with a deletion, skin cells from a number of patients with a duplication. They reprogrammed them into stem cell status. These are called induced pluripotent stem cells, and these are cells that have within them the capability of generating, kind of like stem cells in the fetus, generating any cell lineage in your body. And so they took those stem cells, they took the fibroblasts, the skin cells, reprogrammed them into stem cell status, and then differentiated them into neurons. Turns out that stem cells love to differentiate into neurons. And when they looked at the neurons under the microscope, you see this. So even with a naked eye, which is in fact how this was analyzed with the naked eye of a postdoc, they looked at the images on the left, and you can see significant increase in axonal arborization, in synaptic arborization and axon length. And on the right side, you see in the schizophrenia or in the duplication patients, a significant depletion in synaptic arborization and axon length. So by virtue of having a bunch of postdocs looking at this in the microscope for days on end, this was actually quantitated in the 3 samples on each of those 2 groups. And you can see that there is, in fact, a statistically significant signal as it relates to these image phenotypes that were observed by eye. So basically, this is a very laborious, very small scale experiment that even in that, even though it took a long time, but gave tremendous insight into the pathophysiology of this particular genetic variation. So now imagine that we were to scale this up and industrialize a process like this. And the reason we can do this is because there's technologies today that are each transformative on their own, but together they are a perfect storm of data creation. So at the top is the ability to create disease-relevant cellular systems that really specifically speak to human biology. So if you think about the experiments that we used to do in the olden days, 5 years ago, most of the experiments used to be done in cancer cell lines. And cancer cell lines the DNA is incredibly unstable. So even if they were relevant to human biology at the beginning, which is arguable, they're very less relevant now because every time you passage them, they accumulate more DNA damage. So they're really bad models for other human disease biology. Today, this process of creating these iPS cells that I described earlier can be done for people from just normal genetics. And you can create iPS cells and then generate neurons and cardiomyocytes and astrocytes and hepatocytes and, and all sorts of different cell lineages which encapsulate within them the genetics of people that are more prone to disease and less prone to disease. And you can see how they differ from each other. You can perturb those systems using CRISPR. We didn't used to have the ability to do genome editing reliably and at scale. But now CRISPR allows us to say, okay, what happens if I poke this cell and increase the prevalence of this particular genetic variant that increases the probability of disease? That, in turn, allows us to engineer cells to be healthier or less healthy. And then the last piece of this is the ability to look at these cells not relative to very simple one-dimensional phenotypes like growth rates or the fluorescence of a single marker, but using incredibly high-dimensional content that includes super-resolution microscopy, single-cell RNA sequencing that allows a view of the entire 20,000 dimensional transcriptome, and multiple others that are coming online. So you can really generate and measure cellular systems incredibly robustly, and by using microfluidics and automation, you can actually scale this so that you can do not one experiment every 4 years, like in the paper that I showed you before, but hundreds of experiments in a month. And that all of a sudden provides you with an explosion of data that speaks to how normal and abnormal human biology differ from each other in a way that's driven by actual human genetics. So that gives you lots and lots of data that looks like that. These are actual images of differentiated iPS cells that were differentiated into neurons in our lab, and these are stained with different markers, and you look at this and you say, wow. And how do we analyze multiple hundreds of terabytes of data that look like this? So that's kind of hard, and fortunately, that's why we have machine learning. And I think this is a slide that I mostly use when I give talks to biologists, and they don't know how much progress we've made in machine learning in the last decade or so, ranging from 2005, where we couldn't even tell whether there was a bear in this image, 2012, well, maybe there's a wolf, there isn't, but close. 2014, a brown bear is swimming in the water. It's not actually swimming, but certainly a pretty good caption. And today, where we're actually able to provide interpretations of those images that are actually better than human-level performance. And it's better than human-level performance for tasks that we're trained to perform from birth, like looking at natural scenes and saying, what's there? Yes, and I got this from Sammy, so thank you, Sammy. When you're looking at images like the previous one, we're not trained to look at images like that from birth, and it's really hard to figure out what's going on there. So I don't need to tell you about end-to-end training, so I'm going to skip past this. But I will talk about this, which I think is actually interesting, because it ties into what I'm talking about before. Now, you all know this in this audience, but I think it's worth emphasizing, that in addition to providing good predictive accuracy that you get when you perform end-to-end training, the internal feature representation of these neural network models actually provides an embedding of the data into a lower dimensional manifold that sits in the high dimensional space in which these images usually sit. So if your natural image is 1,000 by 1,000, by the way, ours are 20,000 by 60,000 because super resolution microscopy. But even if they sit in 1,000 by 1,000 dimensional space, the space of real images only occupies a very tiny fraction of the space. And effectively, by training the machine learning model to make certain predictions, you're crafting the architecture of the low dimensional manifold within that high dimensional space and forcing the machine learning to put adjacent to each other in the space things that have similar semantic content. So for instance, these two trucks, even though in pixel space would be quite different, distant from each other, in the low dimensional space would be much closer to each other because the algorithm has to classify them both as trucks. Cars and tractors would be sort of in the same general vicinity because they share some feature similarity in this representation, whereas things like cats and dogs presumably would be relatively far apart. So that representation turns out to be really important for what we're doing, which is why I paused to emphasize this. Okay. So putting those two pieces together, what we're building is what we call the in vitro human platform, which actually creates in vitro or in test tube avatars of human genetics and measures their phenotypes. So if you think about our original set of humans that we started out with, let's say you have a couple of hundred humans in an optimistic scenario for whom you've collected these iPS cells. 200 is actually a lot. The experiment that I showed before, they had 3. So 200 would be a lot. And you put them in the high dimensional space in which they sit in terms of that, those 20,000 dimensional images. Those are, they're going to be scattered stars in the night sky. Not a lot of structure is going to emerge. But let's imagine that what we're doing is we're now using CRISPR to expand each of those points to create a cloud of points around it that really allow the genome to be engineered to be different types of, different levels of disease burden from a genetic perspective. So all of a sudden you have a lot more data. Each of those data points is now a cell with different genes. So you have the original points and you have the fake points, the synthetic human points. And that gives you a whole much larger data set from which you can now learn the low dimensional manifold by observing their cellular phenotypes of the engineered genomes. And now you have this low dimensional manifold on which the original points sit. And in this new space, clusters emerge. So whereas before these were just like scattered points and you couldn't really detect clusters in this high dimensional space, in low dimensional space the clusters can jump out at you, which means that for these complex polygenic diseases, which are almost certainly not one disease, Alzheimer's is not one disease. Autism is called spectrum for a reason. These are different diseases. Often they even reside in different cell types. In Alzheimer's, we have some neuronal phenotypes and some phenotypes that are related to the microglia, which are a different cell type in the brain. So these clusters of distinct disease subtypes emerge from looking at the molecular data. This, in fact, is the transformation that enabled oncology to take this huge leap forward in the last few years, one of the transformations, there were a few others, where we now understand that breast cancer is not one disease. And there's certain subtypes of breast cancer, like the BRCA cancers, that are a lot more similar to ovarian cancer, even pancreatic cancer, than they are to other types of breast cancer. It just happens to originate in the same tissue, so we called it the same thing. It's almost certainly the case that we're calling autism one thing because it phenotypically is sort of slightly similar to each other, but it's clearly different diseases. And so the reason we haven't been able to discern that for diseases like autism is because you can't put a needle in people's brain and start, assaying their brain cells. But with this approach, you can actually do that. And so hopefully the clusters will emerge. And what will hopefully then also emerge is the ability for us to say, well, what happens if I intervene in these cells in the following way? Does that revert the unhealthy to the healthy state? And when that happens, you might have a different intervention for each of those clusters, maybe an intervention even targeting a different cell type. And you can see which of them reverts an unhealthy to a healthy cell state. So fundamentally, the summary of the approach is the following. We start out with human data that allows you the genotype-phenotype association. that gives you a sense of what genetic variants are involved in a particular disease. And that gives the ability to then take that architecture and engineer that into these IPS-derived systems. So we start out from the data, and then we create biological data that mirrors and expands on what we see in the human population. That creates a data set that is robust, at scale, and fit to purpose for machine learning. And by that I mean not just in terms of the quality, but also in terms of the metadata and measuring all the covariates, and I'll talk about that in a minute. And then that massive data set is then used to build machine learning, where you can build phenotypic manifolds and explore the phenotypic space to hopefully make discoveries on what perturbations might actually help revert disease phenotype to normal. And so from this will emerge insights on subpopulations of patients, novel targets that modulate the disease, and the effects of the interventions. So this is a video of the lab. Let me see if it actually plays. Hopefully it will. This is to show you that this is not just a concept. Can we lower the volume, please? Thank you. So this is the end-to-end automation. So you first have to retrieve the cells from the freezer. Then we have the cell culturing system that is actually able to culture 220, this is that one, 220 cell lines at once, and automatically culture them, passage them, and differentiate them into multiple different lineages. The cells grow. They don't grow that quickly, unfortunately, but they do grow. And then you transfer them to the production lab. This is all our lab, by the way, where there are a number of different pieces of equipment. This is the nanoliter dispensing platform. It's the coolest thing because it actually manipulates liquids using acoustic liquid handling. So only sound waves touch the medium. And then you take those cells and you can assay them using either high content imaging, like we showed in some of the examples, but also we have a sequencer, so you can do single cell RNA sequencing, and more assays are coming online. This allows you massive scale. It allows you very rapid turnaround from the time that an experiment is envisioned to the time that the data actually appears. It allows you to do robust machine learning-based experimental design. And everything in the lab is instrumented. There are dozens of IoT devices in the lab that measure temperature, humidity, batch of reagents, the time the machine was last calibrated, and multiple other things, so all the covariates are aligned with each data point so you can correct for that when you're doing the machine learning. So, okay. And so this is just to show you that machine learning is actually happening. This is really early machine learning because this lab is still actually in the process of being completed, but just to show you that machine learning is actually happening. This is basically, you take cells, you treat them with, in this case, 10 different chemicals, and you ask yourself, can we predict by looking at the cells or which chemical was actually used in that experiment. So the cells kind of look like the image on the right. That's what they look like. And this is state-of-the-art. It's a multi-layer perceptron on cell profiler features. This is sort of the standard from the Broad. And you could either do the classification at the level of the entire field of view or at the level of individual tiles. And you can see that you get a significant boost in accuracy relative to the state of the art. And let me just say, for those of you who are thinking, yeah, you just plug this into ResNet, no, we didn't. There is, you know, between here and here, there is a ResNet somewhere in the middle, and this is much better than that. And then, okay, so one point is, this was 3 months of work. The reason why it was 3 months of work is because this is actually like really hard. Manipulating images that are 20,000 by 20,000 is hard. Dealing with a microscope, there's some really nasty, sort of annoying things. Like, for instance, it takes multiple channels. It turns out that the warp on each of those channels is actually different. So in order to actually align the images to each other, you actually needed to do machine learning to make sure that the pixels actually line up properly. But the nice thing about having an experimental lab in-house is that, oh, we don't get the 3 different images to line up because they're warped differently. You just redo the experiment with fiduciary markers, figure out the warping function, then you don't need to do that again. So that closed loop of experimentation is actually really important in getting the quality of the results. So this was 3 months of work. Then we did a much harder experiment, which is you took the same kind of cells and you subjected them not to chemicals, which have a very strong signature, but to CRISPR guides. So you perturb the cells using CRISPR. And within 2 days, this is the nice thing about having these automated pipelines, you basically get results that look like this. Now, you can see that there's still a fair bit of headroom here. 70% is not where we'd like to be, but honestly, you couldn't optimize 96% much further because you've kind of hit the ceiling. Here, we actually have room where we can make the algorithms better. Okay, so the critical part of this is the closed loop between the design, between the creation of the data in this biodata factory and the machine learning and genetics, and they really each feed off of each other in ways that are totally unique and critical to the capabilities that we're trying to develop. You couldn't do this. It was like a off the shelf, it was like, throw things over the wall handoff between the two. So this is one application. But really what we're building at In Sitro is a fully integrated drug discovery and development organization that will use these two together across multiple problems, including how do you design molecules that achieve a particular function? How do you select the right patients for a clinical trial? And many other pieces as you get the drugs closer to clinic. So I'm going to end with a philosophical note, which is why I'm back in this space after after 20 years. If you look back at the evolution of science, you can see that science often proceeds in a sequence of epochs, where in a given epoch, you have a particular discipline that moves really quickly for a certain period of time because there's a new insight or a new tool that's enabled a completely different perspective on that field. In the late 1800s, that was chemistry, where we understood the difference between alchemy and chemistry, that you couldn't turn lead into gold. In the early 1900s, it was a set of advancements in physics about the connection between energy and matter and space and time. In the 1950s, it was digital computing, where for the first time you could actually create an artifact that performed computations that up until that point only humans had been able to do. And then around the 1990s or so, getting towards the turn of the century, there was sort of a bifurcation between two disciplines, each of which started to make tremendous progress. All of you here are familiar with the advancement in the field of data, which is related to computing, but I think deserves to be its own discipline in terms of the combination of computation and statistics and optimization all bundled together. But the other one that many of you may not be familiar with is the huge advancement that happened in biology with sequencing and the ability to measure the transcriptome and mass spectrometry and microscopy and so many ways in which one could actually quantify biology at an unprecedented scale and fidelity. So that happened, but these two threads evolved in parallel. As I said, I've spent most of my career at the boundary rather than the intersection of these two disciplines. But I think this next era that's coming up now is what you might call biological data science or biotech data, or I hope somebody comes up with a good name for it. And that's really the ability to measure biology at an unprecedented level, interpret what we're measuring using the tools of machine learning and data science, and feeding those insights back into biology to engineer biology to do something that's different than it would normally be inclined to do. And that's going to have ramifications in so many different disciplines, so many parts of the world. It's going to have implications in agricultural technology, in engineering better food sources, in environmental technology, in getting maybe bacteria that chew up plastic or consume carbon dioxide. It's going to have ramifications in manufacturing of biomaterials, and it's going to have ramifications in human health, which is where we are. But I think this field is an incredibly incredibly rich field for those of you who are interested in machine learning and maybe want to branch out beyond, e-commerce or whatever, and do something different. I think there's tremendous opportunities in this space across multiple different application areas. Thank you very much.
We have time for a few questions now. There are a couple of mics there.
Hi. So I know that a common issue in biological data collection is that once you collect the data for certain samples, either they die or you alter them so much that reproducibility becomes an issue. Have you seen some advances regarding that issue? Because I feel like before you start hitting them with all these tools, kind of need to.
The lack of robustness of biological experimentation is a huge issue, and one of the reasons why first of all, we're doing everything with automation, because the biggest source of variability in most cases is just human variability in how the experiment is performed. A second thing that we're doing is we're trying to control the environment as much as we possibly can by keeping the humidity fixed, the temperature, and so on. Also, batch of reagents turns out to be really important. So many of you probably don't know this, but cells are grown in fetal calf serum, which is exactly what it sounds like. It's the serum from a fetus of a calf. And different calves are different. So what we did is we actually collected multiple batches of fetal calf serum, mixed them all up into one huge vat so that you could do all of the experiments with one batch of fetal calf serum, so it reduces the inter-batch variability. We're very careful about experimental design so that there's not not all of the experiments relative to one type of perturbation, for instance, are done in one batch, and the different batch is a different experiment, because then you're unable to deconfound them. And then just keeping track of the covariates so that you can at least know when you're overfitting to covariates and then try and correct for that computationally. So that's one of the reasons why we're generating data in-house.
Question from the back.
Hi, fascinating talk. Thank you for that. I was wondering, how do you determine what is the space of perturbations? Is this domain-driven? Do you add perturbations to a diseased genome or a healthy genome? And how does this grow?
Boy, yeah, that's a great question. I guess the good news in some weird way is that the space of genetic perturbations is bounded, and relative to the scale of automation that we have, not that large. There's only 20,000 genes that are in the genome. You can overexpress them or you can repress them. So it reduces the set of at least single gene perturbations. Then you can ask yourself, what about pairs and triples and quadruples? And what about the environmental conditions, like what's in the medium besides the fetal calf serum and all that? So it's finite, but it can get combinatorially large. One of the nice things about having a machine learning model is that you can now start to think about techniques like Bayesian optimization and active learning, so that you can start out with a limited set of perturbations, learn the model, and then feed it back to the lab and say, this is the next set of perturbations that you would want to experiment with. You could only do that if you have the kind of scale and automation that we have in this case, because you need that short turnaround feedback loop, and you need to have control over your own experimental setup.
So the follow-up question was, do you start with a diseased genome or a healthy genome, and does that differ?
Yeah, the answer is we start with a broad range of genomes, because you need to have the comparison point.
Thanks.
Yeah.
Thank you for your talk, Dr. Koller. As you know, disease biology is very complex, and not all systems can be recapitulated in vitro. I'd love to learn about some of the systems that you've been able to model successfully and some of the lessons learned and challenges.
Yeah, so maybe to calibrate expectations, first of all, let me say that we're about a year old. So everything that you've seen here is from the last year, and the lab is only about 6 months old. So a lot of this is very early days, and I can't point to 5 drugs on the market that have used this technology. So answering the high-level question that you asked, you're absolutely right that some diseases can be modeled in vitro and others cannot. This is a place where, in some ways, we are riding a technological tidal wave. Almost every week or two, there is yet another paper that presents an in vitro model for this or that disease. One of the diseases we're working on, it's been in the press, is NASH, and they've shown that you can take an organoid, a liver organoid, and you put it in a medium with free fatty acids, and it begins to develop the same phenotypes that you see in NASH, including inflammation and fibrosis and cirrhosis, and so you can actually build a disease model for that. That didn't exist a year and a half ago. This is a paper from like 3 months ago. So there's things that we can model today, and we're going to focus on those. And in 2 years, there will be more things that you can model because the technology keeps getting better. There's organs on chip now where you can actually connect full organ systems to each other. These are not at scale yet, but in 2 years, they will be. And so over time, we'll be able to tackle more and more diseases relative to what we're able to do today.
One last question from the back.
Thank you for the talk. It was very informative. From what I understand, you have this kind of data augmentation using CRISPR, and I'm not too familiar with CRISPR, so I wanted to know how much of error that causes, and is it propagated into your system as well?
So CRISPR, it's a complicated question because there's multiple variants of CRISPR. There is CRISPR nuclease, which knocks out a gene. There is CRISPR-I, which inhibits, CRISPR-A, which activates. There's CRISPR base editing. Each of those has different error modes and also depends on the guides. So the answer is there are ways in which you could look at a particular CRISPR system and a particular CRISPR guide and quantify the effect of that particular intervention on the system, and then use that as part of the analysis that you're doing when you're doing the machine learning model. And it's not immediately obvious, but there are ways in which you could sort of look and define what the effect of the intervention is, at least at some level.
Thank you.
Thank you very much, Daphne.
Thank you.
The poster session will start now.